Hi, and welcome to this lesson where we're going to be looking at module 3.3 from the OCR A-Level Biology syllabus, which is all about plant transport. So the first thing you're going to need to do is to get these images. You can get them from the PowerPoint that I'm sending you. There's an A3 version, which looks like this, or there's an A4 version, which looks like this. So what you want to do is to first get those images, print them out, and then we can use them to construct our mind map. So if you need to get those images ready, get colourful pens and pencils, then we're going to make a quick start. If you don't have access to the mind map, I'll just disappear for a second so you can take a screen grab. Okay, so where to begin? Well, first of all, let's just talk a little bit about plants and why they need transport systems. So plants need transport systems to move things around their bodies, just like humans do. And the two things that plants move around their bodies is they move water and they move sugar from photosynthesis. So water comes from the roots and needs to be transported up to the leaves and to the rest of the plant. And sugars made in photosynthesis are manufactured in the leaves and they need to be transported to the other cells to provide fuel for respiration. So we've got two systems that work in plants to move these two things, water one way and sugar largely the other way. Plants don't really have a transport system to move things like oxygen around the body because remember they have a low metabolic demand. They don't walk, they don't move, so they don't need a lot of oxygen. So oxygen normally gets around the plant through simple diffusion. So I think the best place to start is actually if we zoom in and we look here. Okay, so we look at this diagram which sort of shows in outline the movement of water through a plant. So let's label. So here we have water moving into the plant. This is the blue arrow here, but it's powered by active transport. So here, mineral ions. Sorry, it's been a while since I've done one of these over half term. I've forgotten. Okay, so mineral ions are absorbed by active transport. So that means it takes energy and then moving from a lower to a high concentration. So therefore we can sort of draw, I don't know, a little few dots in here representing high concentration of mineral ions in here. Because of that, water then moves in via osmosis. So remember this is down a water potential gradient. Out here, these things are sort of soil particles, so I'll do this brown. So this is soil particles. This is a root hair cell. So let's just sort of label that. So this actual cell here, this one here, you could draw an outline around it if you like, is a root hair cell. And of course it has a very high surface area. Also, it would have quite a few mitochondria to provide the ATP for the active transport. So it's quite a metabolically active cell. Let's put that as well, mitochondria. Okay, so the water moves down a water potential gradient, and it keeps going down a water potential gradient as it moves from cell to cell to cell. So we could actually label some sort of water potential values. Let's just kind of make them up. So this might be this cell here might have a psi value of, I don't know, let's say, I don't know, minus 100 kPa or something. And then, you know, if it's going this way, then this one would have to be lower. So this would be like psi minus 150 and so on. So we're moving down a water potential gradient and so on and so on. Now there are three ways that water can move from cell to cell. So let's briefly go over here and label these three ways. Water movement, cell to cell, Okay, so we've got A, B, and C. Pathway A, pathway B. Let's keep the color scheme. And pathway C. Okay, so A, in this case, is the apoplast pathway. So the apoplast pathway is water moving through the cell walls. So remember, a cell wall is really just like a kind of mesh matrix of cellulose fibers, but it's very porous. It's not a solid structure. It's more of a net. So water can move around those cellulose fibers. So there's a pathway there. It can kind of go between the cells, through the gaps around the cells. So that's the apoplast pathway through the cell wall. The second pathway is water can move inside the cytoplasm of cells via the symplast pathway. I'm going to keep using red because orange isn't great. So to write with. So symplast. And this is, so it has to cross the cell membrane. So inside the cell. And it has to cross the plasma membrane, the cell surface membrane. Now, this can be made a little bit easier. If you look at this gap here, these gaps between these cells, sorry, these joins between the cells, these are called plasmodesmata, singular plasmodesma. So it has to cross plasma membrane or via plasmodesmata. And then C equals the vacuolar pathway. So, you know, it overlaps with the symplast pathway. It just happens to go into the actual, across the tonoplast, which is the membrane around there, into the vacuole, and then sort of out, back into the cytoplasm, through to the next cell, into the vacuole, and so on. Okay, so there's 3 different ways the water can move. So if we now go back to this diagram here, when the water's moving from this outer root hair cell further and further inside the root, it can be going any of those 3 pathways. Doesn't matter. Could be going apoplast, could be going symplast, could be going vacuolar. And in fact, a lot of it goes apoplast because that's kind of the fastest way. So via any pathway, but mainly apoplast. Sorry if that's a bit scruffy writing. Via any pathway, but mainly apoplast is here. Now that all works fine until it reaches this special layer of cells which has something called the Casparian strip. So this here, I'm going to label it down here, is the Casparian strip. So this is a ring around every single cell. So imagine this cell here is a 3-dimensional kind of block, but there is a ring around that cell. I need a prop here. I'm going to use my hard drive. Okay, so here is a hard drive. Let's imagine this is a plant cell. It's kind of 3-dimensional. The Casparian strip is a bit like a ring. Oh, my hand is not quite big enough to fit around it, but imagine a ring just kind of fitting around that cell like that. So that ring around the outside of the cell blocks the apoplast pathway. Okay, so now the water can't go through the cell wall because there's this ring blocking it, and it has to go inside the cell via either the vacuolar or the symplast pathway. So the Casparian strip, let's just make a little bit of a note here. So it's a ring around all the cells in this layer, and it's impermeable, and it's made, I don't think this is on the syllabus, but it's made of something called suberin, which is just it's an extra a biological molecule that kind of just packs in all the gaps in the cell wall to make it impermeable. So the purpose of that is that we make water move via the symplast or apoplast pathway. Now there's another key process that happens. So here, we could do, actually, let's draw another arrow in red, because there's a process that's happening here, which is the same process as the previous red arrow we drew, which is there's active transport, okay? So that's what's happening here, this thing here. So more active transport of mineral ions into this central part of the root. So in this central part of the root, we call this the vascular bundle, and this is where the xylem are going to be. More active transport of mineral ions into vascular bundle. So what's the point of that? Well, you pack more ions into the vascular bundle, which means, again, we lower the water potential, the psi value, and water moves via osmosis. So then this water moves via osmosis here. So that's this one. I'm just going to label that up here. Water follows via osmosis. Okay. This generates actually a pressure, okay? So there's loads of mineral ions and water kind of packed in the vascular bundle. So then this generates a high pressure environment in this space here. I'm going to kind of do that with a little kind of highlighted blue. And then I'm going to try and keep the same vague color. And I'm going to write that. I'm going to write root pressure. Brackets high. It's high pressure in there. Okay, so that root pressure is part of what pushes water up the stem towards the leaves where it needs to go. But there's a lot of other factors as well, drawing the water up. Okay, so now let's take a look at what that root and what that movement of water through the root looks like in a cross-section of a plant root. So if we look over here, okay, this area now, this is a cross-section of what a root would look like. So it's actually not the full root, okay? So let's kind of add to it, okay? So yeah, where am I going to add? Let's add it down here, okay? So this imagine the root is a circle, okay? So like this, okay? And actually, on the edges of the root, we've got the root hair cells. So we've got all these root hair cells like this, kind of going around the whole root, like that. Okay, we can add some more over here. Those are the okay, so let's just label that root hair cells. So we know that water gets absorbed via the root hair cells. It's osmosis, but powered by the active transport. So water's coming this way, this way, this way. Okay, in, moving via osmosis, following the water potential gradient we talked about here. And it goes in here. Now this is the layer here where the you can see this layer of cells right there. That is the layer where there is this Casparian strip. So I'm actually going to, this here is the layer of cells that contains the Casparian strip. Now we also have to give it another name. Not too bad. So this is the endodermis layer. So it's pointing to this layer here. Can you see? Whoops, that's a bit too big. But that is called the endodermis. And it contains this Casparian strip. Link that over to there, okay? So in from that, we have all these cells here are just kind of random packaging cells. There are two types of cells. They're called the sclerenchyma and the collenchyma. I always forget those. And together we just call it the medulla. So these cells here, okay, so this is the medulla here, and on the left, it's just the cells that are kind of here, okay? So those cells don't do anything, they're just kind of packaging material. The cells that are really important here are the xylem and the phloem cells. So let's label the phloem in green, okay, phloem cells. We're going to look at phloem in great detail later, but remember the phloem cells transport sugars, and we've got the xylem cells that transport water upwards. I tried to do green for sugar because sugar is made in photosynthesis, and I think of green when I think of photosynthesis, and blue for water for the xylem. I'm also just going to highlight that a little bit. So first of all, Let's do the xylem in blue, because that's what transports the water. And we can put the phloem in green in a sec. Okay, so that's the xylem, and then the phloem here in green. Kind of in this kind of just amongst, it looks like a starfish basically. The xylem looks like a starfish with little blobs of phloem in the kind of armpits, I suppose, of the starfish. If a starfish does have an armpit, yeah, then that's where the phloem is. Okay, and then the packaging cells, I'll just leave blank. This medulla is around the outside. Now there is another thing there which you need to know. There is a layer of meristem cells just underneath the endodermis as well. It's called the pericycle, but let's also just talk about that. So endodermis contains a Casparian strip, meristem underneath, and that's called the pericycle. Pericycle, yeah. And the other thing is that the endodermis cells, again, lots of mitochondria because they're very active. They do lots of active transport, pushing the mineral ions into the vascular bundle so that water follows. So they have a lot of mitochondria. They're very active. Active cells, mitochondria. Okay, so that is the cross section. Let's make sure we understand what that is. And then outside the endodermis, this is the cortex. Okay, so this is outside the vascular bundle. So that's kind of this area, sort of here. Medulla's inside the endodermis. So this is a cross section. Root cross section. Okay, so far we've talked about how the water gets into a plant via the root hair cells, how it's moved into the vascular bundle via the action of the Casparian strip, and now we're going to talk about how water moves up the xylem vessels. Before we do that though, there's one thing which I kind of should have stressed a bit more. The Casparian strip, what's it about? Well, I've already said that it forces water to go through either the apoplast or the symplast or the vacuolar pathway. It blocks the apoplast pathway. But why is that important? Well, it's really actually about the mineral ions. So without the Casparian strip, if you actively transported mineral ions into that vascular bundle across the endodermis, then those mineral ions could kind of leak back out through the cell walls because it's kind of like a gap. So that would mean that you couldn't have this concentration difference where you have more mineral ions in that vascular bundle, which draws water in via osmosis. Okay, so it's actually really to make a sort of watertight separate compartment on the inside of the root, that's the vascular bundle, where you can generate the root pressure. So we already talked about root pressure, and so that's one part of pushing the water up the xylem. But now let's talk about the xylem themselves. What are the adaptations of xylem tissue that mean that they are very good at sort of drawing the water up all the way to the top of the tree, if it is a tree, to the leaves of the plant. So we're going to look at this diagram down here, which shows both xylem and phloem tissue. So I'm going to talk about the xylem tissue down here. And later on, I'll sort of leave a little space here for the phloem tissue over here, but that's going to be a little bit later. I'm going to try and keep my xylem stuff down here. All right, so the first key thing about the xylem is they're dead, okay? Now, you may be sort of wondering, how do you build a dead cell? Well, they don't start off dead. So xylem cells, when they're growing and developing, they're alive. And once they kind of reach the shape and structure that they need to be, then we have a final stage where you have something called lignification. That's adding of lignin into the cell wall. And that really is what kind of kills the cell, because once it becomes completely lignified and impermeable, then it can't really live. It's dead. So dead cells. Lignin in cell walls. So the walls have added lignin. Lignin is basically, it's a protein, and it's basically what wood is, okay? So wood is lignin, essentially. So the xylem get hardened by lignin. Now, lignin in cell walls doesn't have to be fully lignified, so let's do a little subheading in here. So it could be a pattern. So it could be spiral. It could be sort of full. It could be what's called annular, which basically just means rings. And even if it is fully lignified, the whole sort of xylem vessel is fully lignified, you can have parts of the sides of the cell wall which aren't lignified. These are called bordered pits, and they allow water to move out. So even if it is full, you might have bordered pits, and that allows lateral movement out of the xylem. So let's add in a little. Actually, I'll use red just because it's on blue. So let's say you'd have water moving up here, up the xylem like so, and then here, look, there's a little gap there. Okay, it might come out. Okay, so this could be a bordered pit here. So that allows water to keep moving up the xylem, but some of it to exit at different parts to supply the tissues. Okay, so that kind of shows lateral flow out. Okay, because of course if you have a tree, you might have a branch here, and then more up, and another branch here. So some water needs to come out into this branch, and some water needs to come up into this branch, and so on. So that's the main thing about xylem. Now, the fact that they're dead and the lignin in cell walls means they are tough, essentially straw. Okay, they're basically like a straw. They join end to end, so the cells fuse. Okay, so dead cells, lignin in cell walls, and I didn't really state the obvious here that this is strength. Okay, lignin in cell walls give strength. Now, if the tube is fully lignified, it's strong, but it might be brittle. It might snap. Okay, so spiral gives it strength, but it gives it some flexibility, and the same thing for annular. So spiral might be flexibility. Okay, right, what else? So dead cells, lignin in cell walls. Cells fuse end to end. Okay, so they, one cell and the next, the end plates fuse to form a continuous tube. Continuous tube with no cell contents, nothing at all, and no divisions between individual xylem vessel elements which fuse together. So the reason that this is so important, this continuous tube, is then it allows the properties of water to kind of take over. So you may remember that two properties of water that we looked at in the sort of molecules mind map were, so I'm going to underline this continuous tube here, and I'm going to put H2O. We've got cohesion, and which is together, water. I'm kind of writing this in shorthand. Cohesion is the force that attracts water molecules together. They are cohesive. And we also have adhesion, which is how water molecules stick to the walls of the xylem and to other things, okay? So now we have this continuous tube. Cohesion and adhesion can kind of allow the water to move up the tube. And this is powered really by things that are happening in the leaf. Water is evaporating out the top of the leaf. So this pulls water molecules out the top of the xylem, but because all the water molecules are kind of sticking together through cohesion, when you pull one out the top, it draws the water molecules from the lower in the xylem up like that. Okay, so that's cohesion. And adhesion also is the water molecules stick to the sides of the xylem and kind of almost crawl up the xylem vessel via capillary action, which is another key word. So cohesion and adhesion together in the narrow continuous tubes give rise to what is called capillary action. Okay, so I think that's everything about the xylem. Yeah, the other thing, so lignin is strong. It also prevents the xylem vessel from collapsing in on itself. So it keeps it open as a tube so that water can flow. So that's the xylem features. So this is the diagrammatic representation. Here's an actual microscope image where the lignin has been dyed red here. So here we can see the kind of ring-like, or it could be spiral, I'm not sure, pattern of the lignification of this xylem. So, okay, so this point here is really water flowing in xylem. In stem. Okay, so that is the xylem flowing in the water flowing in the xylem in the stem. Right, but we need to, again, recognize the shape of a cross section. So let's go and look at that. So here is a stem cross section. Now this would be a stem cross section. Not every single plant's going to have exactly the same stem cross section, but this is a stem cross section in not a tree, so sort of like a, I don't know, maybe like a sort of a semi-woody plant. Okay, I think it's called, a non-woody plant. So something like a green plant, bushy, which isn't like a tree with a full-on trunk. In trees with full-on trunks, there's a slightly different structure because all the vascular bundles fuse to form a ring, which is why you can count tree ages by looking at those rings that get laid down every year. So in a non-woody plant, we have discrete vascular bundles. So that's what these are here. So let's actually just highlight one, that whole thing there, and we'll just label it. We'll just say that is a vascular bundle, okay? And in fact, in your textbook, there is a further diagram of a vascular bundle, which I am going to try and draw now, but maybe you could draw it better than me. It's on page 209 in the middle, and it kind of looks a little bit like this. That's the outside layer. This would be a tissue plan, okay? So I'm just going to draw the kind of the tissues there, and then we have the sort of the phloem like that, and then we have the cambium, which is a very thin layer like that, and then we have the xylem on the inside. Okay, so that's kind of, this is a more high power, well, this is actually a tissue plan, but zooming in a little bit more. So that diagram is shown on page 209. If you wanted to add that as well, you could. So let's just quickly label that as well, and I'm going to do it in blue. Yeah. So, okay, we got this bit. This is the sclerenchyma. Then underneath that, we've got the phloem. So in plants, in the stem, the phloem is closer to the surface. And that goes for non-woody plants and woody plants. Down here, we've got the xylem. What's in the middle? That is something called the cambium, which is basically meristem cells. Okay, so cambium, meristem. Now we will learn about this more in year 13. We learn about plant hormones, and you need to know a little bit about where the different meristems are in plants and how that causes plants to grow, like out and up and that sort of stuff. Okay, so now let's label this diagram, the slightly zoomed out version over here. So this outer layer is the sclerenchyma. Then we've got the collenchyma. So actually that's the even further outer. That's like around, that's on the very outside. So that's kind of the protective layer on the outside of the plant. We've got a bit of cortex in there, which is not in the vascular bundle. It's between the outer collenchima, and then the vascular bundle. Then we have the phloem, and then we have the xylem, and then we have this bit down here is the medulla. Okay, like that. Right, let's try and keep the same highlight as I did down here. So I'm going to do blue for xylem. So I'm going to try and colour them all in. So xylem is on the inside. I won't colour this one, which I've highlighted in full, because that's the whole vascular bundle. So there's the xylem, all those little bits. What else? Now we need to do the phloem. Oh gosh, this is going to be difficult to do. It's this middle bit here. Yeah, there we go. That's the phloem. And I'll do the collenchima, sorry, the sclerenchima in yellow here, which is that kind of packaging. And of course, remember in this slightly zoomed out version, you can't, there's not enough room to label this cambium down here. Let's also just, on my zoomed in version, let's do that color code as well. Remember, we don't color in biological drawings normally. It's just so you can see where everything is. Yeah, and we'll just do that like that. Okay. All right. So again, you need to recognize this kind of pattern of where the xylem and phloem are in different parts of the plant. How are they arranged in the root? How are they arranged in the stem? Okay, so we've covered water flow now from the roots into the xylem, up the xylem. Now let's move on to the leaves. Okay, so here we are in the leaf. Now we're actually going to start by looking at this diagram here, which isn't in your text, but it's very similar to the one which is on page 214, which is a diagram of the leaf structure. So first of all, let's just remind ourselves about the different parts of the leaf structure, because we do need to know all the different features. So at the top here, we've got a waxy cuticle. This is the upper surface of the leaf that I'm labouring. Waxy cuticle stops water loss from this upper surface. Stops water loss just kind of out of the cells. So it's a barrier. Then we've got this layer, these cells here. This is the upper epidermis. Now there's some overlap here with the mind map you did the other day. So you'll hopefully always already remember that these are basically transparent cells. They haven't got any chloroplasts, and they basically they form a barrier layer, and they let the light through. And let's add a little bit of light coming in. They let the light through down to the main photosynthetic cells, which are here. Then we've got this layer. This is the photosynthetic layer. We've got the palisade mesophyll. Again, won't go into too much detail because you've already done a kind of bit of work on this, but we've got the chloroplasts are tightly packed. See these little dots here? Those are chloroplasts. Tightly packed with chloroplasts. Also, the central vacuole pushes the chloroplasts to the outside of the cell where they are sort of closer to the incoming light so they can capture the maximum amount of light. I'll just write underneath there, here, photosynthesis, all right? These are the ones. So these cells are going to be providing sugars for the plant. Okay, we haven't really talked about the phloem and sugar transport, but this is the source, the photosynthesizing cells in the leaf. Then underneath that, can you remember what it is called, this one? I hope you can. It's the spongy mesophyll. And of course, we've got all these lots of air spaces. It's spongy, like a sponge. In a sponge, you've got bits of whatever the sponge is made of, the stuff, and then you've got little tiny bubbles of air all over the place, little pockets. And these are interconnected. Of course, a leaf is a three-dimensional thing. It's not just a slice like this. So these air spaces here, are all connected through sort of weaving tunnels and stuff. It's a network, a spongy network of the air spaces. I'll just label that air space. And here is where we've got the vascular bundle. So I'm actually going to kind of draw a ring around that there. And we shall, what should we label it? Vascular bundle. Now in the vascular bundle, you've got xylem and phloem. Typically the xylem, I'm just going to write X for xylem is on top and P for phloem is on the bottom in the leaf there. If you were looking at higher power, you might be able to tell the difference because xylem will tend to be slightly larger, rounder, and hollow than the phloem, which are a little bit smaller. Okay, so then we've got this. This is the key thing. This is the stoma. Stomata? This is the stomata. One is also a stomata. Stomata for, is it one? Yeah, I think stomata is both singular and plural. Stomata, yeah. So this is the stomatal pore, let's call it. And these two things here are guard cells. So the key feature is that water, and I'm going to try and do a nice light blue. Let's do a different color. Water moves out of the xylem here, down a water potential gradient, so it can move, into these cells where it's needed for photosynthesis. It can move through these cells, and it will evaporate. Okay, it will evaporate into this. It's a bit difficult to see here with this color, but it evaporates into this airspace. So this airspace here, if you focus where my pen is moist. It is humid. It is packed with moisture. That moist air can diffuse out of the stomatal pore into the environment, and then it's maybe blown away by the wind, washing over the leaf. So it's this process of evaporation and then the diffusion of water out of the stomata that powers the whole process of transpiration. This provides the pull. So we've got the pull of the water leaving the stomata. We've got the cohesion and the adhesion in the xylem vessels. We've got a little bit of root pressure coming from the roots. And all those things together kind of equal transpiration. So let's label those 2 or 3 key points here. This diagram is in your book. So we've got here, this is water moves out of xylem. Remember, this could be through something like some bordered pits, because of the lignification, it has to have a way out to lateral movement. And then the water, when it's moving from cell to cell to cell, this is osmosis, because we're talking about the movement of water from a high water potential to a lower water potential. Here, this arrow is evaporation, okay? That's kind of key. So I'm going to do that in red, because it's something which we might go wrong. Evaporation, because it's evaporation of water from the surface of the cell into the air space. Now we've got water vapor, a gas, and so now we're talking about a gas. It's no longer osmosis, but this is diffusion out of the stomata, okay? So just a slight difference there, evaporation, diffusion, and osmosis. We've got to use those words at the right time to describe the right process. So that is transpiration. This can be regulated and stopped by the closing of the guard cells. So the guard cells here, let's just put a little note underneath, can close pore in times of water stress. Okay, so if the plant senses that it is running out of water, then the stomata will close. And if the plant senses it's got enough water and it wants to be photosynthesizing, there are also mechanisms that kind of mean that when the plant starts photosynthesizing, that sends rapidly, it sends a message to the guard cells to open the pore. We'll investigate that a little bit more in year 13. So here's a little overview animation, just so you can kind of visualize that whole process of transpiration and the transpiration stream once again. So here we can see at the top that the whole process is powered principally by the evaporation of water from the leaves out of the stomata. Let's zoom in. Let's look back at the root. So here we've got the root hair, water moving in via osmosis, down a water potential gradient. Now the cells are interconnected, so water is moving through the plasmodesma or plasmodesmata via the symplast pathway mainly, or the apoplast pathway. Sorry, the apoplast pathway is the main pathway, and that's through the cell wall. Now that carries on through the symplast, down the apoplast, all the way till we get to this endodermis layer. The endodermis layer has the Casparian strip, which blocks the apoplast pathway, and there is that protein called suberin that kind of makes that cell wall impermeable. This allows the active transport of mineral ions into the vascular bundle so that there is a high concentration of solutes there and a low water potential, which means that water will flow into that vascular bundle via osmosis. This sets up a bit of a root pressure. And now when we're talking about root pressure, that kind of pushes the water up a little bit. But the main thing that pulls water up is really cohesion of water molecules. So here we see this nice animation showing that water molecules are held together via a hydrogen bond network. So as water molecules are removed at the top of the transpiration stream all the way up in the leaf, it pulls water molecules up from lower down in the plant. So we can follow that transpiration stream all the way up to the xylem and all the way up the xylem and into the leaf. Now in the leaf here, the xylem loses water down a water potential gradient into the cells in the leaf. And eventually that water will evaporate into the air spaces within the leaf, and it will then leave via diffusion out of the guard cells, which is the whole thing. This bit is called transpiration, and the whole thing is called the transpiration stream. So as I said, this whole process, transpiration, is super, super important. Transpiration supplies water for photosynthesis in the cell, in the leaf cells, because the leaf needs water. It's one of the raw materials of photosynthesis. Supplies water to maintain the turgor pressure, to keep the cells kind of inflated with water throughout the plant. And it also cools down the leaves when they're in the sun. This evaporation of water and sort of diffusion out the stomata cools down the leaves as well. So transpiration is very, very important. And we can measure transpiration, which is technically speaking just the loss of water vapor from the leaves. Let's just label that. We can measure the rate of this using something called a potometer, which is this device here. Okay, so to investigate transpiration, we use a potometer. So to use a potometer, first you need a piece of a branch or a piece of a plant that you want to investigate. Now, here's how we set this up. First of all, well, there's kind of two types actually. There's one which is a bubble potometer and one which is a mass potometer. So let's just have a little look at this one. We're not going to talk about it in detail, but this mass potometer basically works by measuring, let me go back. This mass potometer here basically works by measuring the mass of water lost. Now here, there's a little bit of a layer of oil across the top of this beaker. There's water here below. And basically, as water moves up the xylem and out the leaves, the mass is going to go down. Now, the water can't evaporate through the oil layer, which is essential if your results are going to be valid. So we just measure the mass lost over time. Now, the bubble potometer is a bit better. And here we go. Let's look at this bubble potometer. So, first of all, if you want to compare different rates of transpiration, you're going to need to use the same cutting several different times, because the different cuttings are going to have different surface areas of leaf and different amounts of leaves and so on. So we're going to need to use the same piece of leaf and just repeat the experiment several times. Now when we cut the branch, which we're going to investigate, it should be cut with a sharp knife underwater. Okay, that is so we don't get any air bubbles introduced into the xylem, which would block the flow of water up the xylem. If you get a bubble in the xylem, you can't have that cohesion of water molecules going up it. So we've got to cut it underwater. Then we have to place the newly cut shoot into the potometer, all whilst it's underwater. Again, this is just to stop the bubbles getting into the xylem. So this whole thing is now filled with water. Now we're almost ready to go. So the leaf has started to do some transpiration. I'm just going to back it up there for a second. The leaf has started to do some transpiration, and therefore air is drawn in. Okay, air is drawn in here. Watch very closely right here. Air is drawn in, so you let a little bubble go in, then you put it in the water. Then you just track how fast this bubble is moving. You do that on a scale, and you watch the bubble go. You time it, maybe 10 minutes, maybe 20 minutes, and you record the distance the bubble moves. So you probably wouldn't sit there and watch it for every single second. You might look at it every 3 minutes for 30 minutes. Sounds about sensible, okay? Then you could sort of plot a graph of this. So over time, you watch the bubble move as it moves along, and you record the time and you plot a graph. So the slope of this graph is going to become the rate of transpiration, basically, okay? Now, what you might do is then you might use this as your reference point. That's basically your control, how much transpiration happens with the branch as it is. But now let's change some factors. Let's see what happens if the leaf is illuminated with a bright light. Now, as a control here, we place a heat shield. Okay, so this could be a piece of perspex, which is specially designed to block infrared radiation, or it could be kind of a small, almost like a small fish tank with water in. Okay, so the water basically absorbs the infrared and it allows the light to go through onto the plant. So we're not changing two factors, light and temperature, we're just affecting the light. So now let's run this experiment again with a higher light intensity. And we can see here that got a bit of variability. The data's kind of going up and down a little bit there. But basically, it's going to look like we've got a higher rate of transpiration. Now, the reason for that, as I sort of mentioned earlier, in year 13, there is a direct link between photosynthesis and the stomatal opening. So it just means that the stomata will be more likely to be open, therefore more transpiration. Light energy also can directly kind of help the evaporation of water. All right, what about air movement? Let's put a fan next to this piece of branch here. Now, the air is going to blow any moist air that has recently come out of the stomata and is just sitting near the surface of the leaf. The fan is going to blow it away. So this is going to increase the rate of transpiration because it means that humid air does not sort of cluster around the leaf. It's going to maintain actually what it's doing is maintaining the concentration gradient in terms of moist air into and out of the leaf because it's blowing away the moist air out of the leaf. Okay, so that's also going to increase the rate of transpiration, not quite so much there. What about humidity? What if it's a humid day? Now, to model this, we could just place a plastic bag around the whole branch, the whole setup. This means that water vapor, when it gets into the plastic bag, it can't be blown away by the air. So the air in the bag becomes quite humid. So does that affect the rate of transpiration? Well, yeah, it clearly does. And this time, it's going to reduce the rate of transpiration because it means the difference in humidity in and out of the leaf is less. So there's a lower concentration gradient in terms of the moisture in the air from inside to the outside of the leaf. Okay, so we can summarize that like this. Okay, ta-da! So let's add this diagram to the mind map. Here it is. Just move it across, and then now let's talk about a few of the key points from that potometer experiment that we need to remember. Okay, so red here was high light intensity. Blue here was windy, replicated with a fan. Black was control, and green was high humidity. And humidity lowers the effect of lowers the rate of transpiration. Windiness or air movement increases the rate of transpiration, and high light intensity increases it a lot. Key facts about the potometer use there was same branch, cut underwater, let's just say cut plus set up underwater. And the reason for that was really kind of no bubbles. In the xylem, that is. But you do track the movement of this bubble here. By the way, you can also, if you know the distance the bubble has moved, you can calculate the volume of water that has been lost. Now, how do you do that? Well, if you know the diameter and the radius of the capillary tube, you can work out the volume of water that has been lost out of that tube. So it is the distance in millimeters times pi r squared, which is the area, because remember the capillary tube is circular, cylindrical, so that's the area inside times the length. So it's basically the volume of a cylinder. Okay, so almost finished on transpiration. Now we're just going to look at how do two different types of plant kind of work with transpiration to, because they live in weird or extreme environments. Okay, so we're just going to touch on these two different plant adaptations. We're going to look at xerophytes and we're going to look at hydrophytes. Now, it's fair to say that plants, normal terrestrial plants, typical plants, also have adaptations to stop water loss. We've kind of already looked at those up here with the leaf, for example, the waxy cuticle. But if you want to add more information here, you can. On page 218, you've got a bit more information about terrestrial plants. So xerophytes are plants that live in arid environment. Okay, so here is a xerophyte. Cactus is a xerophyte, or marram grass. Okay, so this one, the one that we're seeing here, is marram grass. And a cactus is another one. Now, I think, hopefully, as A-level students, you're familiar with a cactus, okay? Cacti don't have leaves. That's the key thing. They minimize water loss by losing all their leaves. So where do they do photosynthesis? In the stem. The stem is green. They protect the stem with spikes from herbivores that want to kind of eat the plant and get the water inside. And the stem itself is kind of very spongy and can store water. But a marram grass is a slightly different thing. So first of all, here's a picture of marram grass for real. Okay, looks pretty much like grass. But what happens is the leaf, okay, imagine my hand is a blade of a leaf. The leaf is not flat, it is curled up like this, okay? And that's what we see here. So the outer surface of the leaf is on the outside of the curl, and the inner surface is inside there. Okay, so this is outer surface, which has got the standard waxy cuticle. So now the inner surface is here. So the inner surface is all curled round, and this means that any water vapor that is kind of coming out of the stomata, and the stomata are actually way down in these sunken pits here. Okay, so the stomata are way down in there. So water vapor has a long way to go to get out kind of in the leaf. And the water vapor, I'm going to change to a little slightly darker blue, so we can sort of picture, water molecules knocking around here as water vapor. So they kind of get trapped inside that space, and it means that the water vapor doesn't diffuse out and away from the leaf so quickly. So this reduces the concentration gradient in terms of the moisture of the air between the kind of inside of the leaf and the outside of the leaf, because the outside of the leaf is this protected environment, protected from wind, and which keeps some of the humid air in. So marram grass reduces the loss of water, and the stomata, I'm just looking where they are in pits in the lower epidermis. Okay, so what are the key things that we could say here? So stomata in the pits, and the lower surface of leaf is rolled Plus it's extra hairy. Hairy pits, essentially. Nice. So yeah, marram grass got hairy pits, and yeah, and it's got stomata there as well. So that all prevents so much water from being lost. So marram grass typically lives on sand dunes and stuff. The outer surface also has, an extra thick waxy cuticle. Okay. Extra waxy cuticle. So that's a xerophyte. What's the opposite of a xerophyte? Zero, I think, is Greek, by the way, for dry. So xerophyte means dry loving. The opposite of a xerophyte is a hydrophyte. Okay, can you guess what this one loves? Well, hope you did. It's water. So water, phyte, likes it. Okay, water-loving plant. Now, water-loving plants, they don't really care about transpiration because they live in water. Okay, so water-loving plants, there's an infinite amount of water, essentially. If you're sitting in a lake or a river, you don't need to worry about losing water out of your leaves because there's just loads of water everywhere. So hydrophytes don't really have to conserve as much water. Now, one thing about, let's just actually draw a little plant here, okay? All right, so I'm going to do a cross-section of this plant, okay? Bear with me. This is the surface of some water. On it, we have a lovely water lily, okay? Water lily, like that. Now, water lily has a stem that goes down like that into, let's say, a pot, okay? This is actually a model of my mum's pond, okay? She's got a pond. It's great. I spend hours looking at it and trying to find out all the nature that's inside of it, and it's got water lilies in it, okay? So the water lilies are planted in the pot in the bottom of my mum's pond, and they've got these big stem things like this that kind of curl up to the surface, and the leaves are floating on the surface. So one thing that the leaf has to do differently is that actually stomata on the upper surface, which is weird because normally they're on the lower surface. So the reason that is essential is because the lower surface is water. So to do gas exchange with the air, to get carbon dioxide from the air, the plants need to have the stomata on the upper surface. So that's one key thing. Also, the leaves and the stem need to float up to the surface. So that's what these things are, okay? These are air spaces. And they cause the leaf to float. So it helps for buoyancy, and it keeps the leaves afloat. And it also allows oxygen to diffuse down those. These kind of air spaces are actually channels for air, for oxygen to diffuse down the stem and to get to the roots, okay? Because sometimes the water may not have enough oxygen for the roots to kind of continue their metabolic processes. So air spaces, two things here. Oh gosh, can I spell this? Buoyancy is a tough one. Buoyancy plus O2 diffusion. Okay, so that's hydrophyte, that's xerophyte. This is my mum's water lilies. And this is the bottom of the pond here. They're just in a little planter. Okay, so that's everything about water movement through the plant. We're so nearly done, but we've just got one little bit more to do, which is about sugar movement in a plant. Okay, so we already said, if I pinch out slightly, that sugar is made by photosynthesis up here in the palisade mesophyll, mainly of the leaf. How does it get everywhere else? Every single cell in a plant needs to do respiration, but the sugar is only made in the photosynthesizing cells. So how does sugar move? Well, it moves in the phloem. So let's go down here and talk about that. First, let's talk about the adaptations of phloem cells. So phloem, unlike xylem, if I move across here, is alive. I say just because the cells are kind of clinging on to life. Living cells. Why are they clinging? Well, because there's nothing in them really, okay? The nucleus, the mitochondria, all that stuff that's normally in the cell has been cleared out in their development process. Unlike the xylem, they don't go fully and completely die, but they, there's not much in the middle. So living cells, most of contents have sort of been cleared out. How does it say it in the textbook? Yeah. Just to say most of content's cleared out. And let's say just a little cytoplasm left. So we can see that here, okay? There's not much in there. There's no nucleus, there's no mitochondria, it's just a tiny bit of cytoplasm. So this leaves room for mass flow. There's a key word, okay? Mass flow. We'll put it in capitals. Mass flow. The other thing that helps mass flow is that the phloem cells, the barrier where one phloem cell meets the next is like a sieve. So it's called a sieve plate. The end is perforated. It's got lots of little holes, many plasmodesmata to the next one. So this is the sieve plate here. So sieve plate between cells. And there is that, okay? Now, the phloem cells have to be alive because there are metabolic processes that have to happen for them to do their job. And those metabolic processes are largely done by something called a companion cell. Okay, so this here is a companion cell to the phloem cells. Without the companion cells, the phloem cells would die. Okay, so the companion cells and the phloem cells are connected by these plasmodesmata. So the companion cells are kind of doing all the protein synthesis, all the respiration, all that stuff to support their neighboring phloem cells. So all these cells here, actually around, would be companion cells as well. So I'll label that next to the phloem. So what do the companion cells do? The companion cells help load and unload sugar into the phloem where it's being made in the leaves, and then out of the phloem where it's being stored, let's say in a underground sugar storage organ like a potato, stores sugar as starch. So first of all, let's look at the loading. So the loading process of sugar. Okay, so let's look at this. Companion cells loading. How do the companion cells stuff sugar into the phloem? Well, it's a bit of an odd process. It's kind of a bit of a roundabout process. First thing, H plus pumped out. Okay, seems strange. We start with pumping something out. Hold on a minute. This creates gradient. Okay, so we've got lots of H plus outside, not a lot in. Then H plus flows back in. Okay, it flows back into the companion cell. But this involves something called co-transport. So every time an H plus ion wants to come back into the cell, it goes via a carrier protein and it carries with it sugar. So H plus can only enter if it brings sugar with it. So this is co-transport. Co-transport of H plus and sugar together. Is it sucrose together? Sucrose. Okay, so now we've got lots of sugar here. Then the sugar, because this is high sugar here, some of the sugar is just going to move into the phloem via the plasmodesmata. So sucrose moves into phloem. So now we've got loads of sugar in here, loads of sugar in the phloem. What happens when you've got lots of sugar in one place? Water follows via osmosis. Okay. this sets up kind of a pressure in the phloem at the source. So this loading is happening at the source. To fully understand the whole bit of the phloem, we've got to just move across here and also look at the sink. So this is the source, okay? We've talked about this specific details of the loading process. So this red arrow here shows sugar being loaded into the phloem. And then we can see that water follows, these two blue arrows. So water follows like that. It also follows from xylem, which typically run next to the phloem. So this is H2O moving from the xylem, so this is xylem, into the phloem. So this generates a high hydrostatic pressure. There's physical pressure there. Now what's happening at the sink? Okay, this down here is the sink. In this diagram here, we've got a beetroot. Okay, so a beetroot has leaves, that's where the photosynthesis happens, and the beetroot is underground. So the soil is kind of here. The beetroot is buried, and it's where the beetroot stores its sugar for the winter, for example. So in the sink, we have active unloading. Let me check that. Yeah, so in the sink, we have active unloading of the sucrose. You don't need to know the full details of this, like the biological mechanism, but there's active unloading of sucrose at sink. So what happens there? Well, again, that means that we're going to decrease, we're going to increase the water potential here. You can see this diagram here, that we kind of, the sugar is being moved out. But crucially in this one, it's then probably going to be converted to starch. So it won't have much of an osmotic effect because it's insoluble starch. So it's moved out, converted to starch quite quickly. So that means that the water potential here is now quite high. And remember, now we're kind of down in the roots. And remember that also at the same time in the roots, we've got mineral ions being pushed in there. So that's why this diagram shows water moving from the phloem back into the xylem, because we're helping to build up that root pressure, partially caused by the mineral ions being pushed in there. So what's going on to move the actual fluid in the phloem? Well, we've got water pressure sort of building up here, water in to the phloem at the source and water out of the phloem at the sink. This causes something called mass flow. So mass flow is right here. I'm going to label that mass flow, caused by the kind of water pressure, so the water going in at the source and coming out at the sink, which is also caused by the sugar. Okay, so that's the phloem. All right, so I think we've looked at everything there. So I'll just zoom out so we can kind of get the whole view. Now what I'll do, because there was one or two more animations I wanted to kind of put in, but I'll leave them up to you whether you look at them. What I'll do is I'll link in my favorite 2 or 3 animations about transpiration and the phloem underneath the video in the description. So if you want to have a look at that, you certainly can. But that is plant transport. Very briefly, I'm going to summarize in about 20 seconds. Okay, xylem, dead cells, lignin, could be different patterns like spiral or ring thickening. Phloem, living cells. Most of the contents is gone. There's a little cytoplasm in there, and they're kind of kept alive by the companion cells, which have a role in loading sucrose. Overall, water moves from the roots through the cortex into the vascular bundle, where there's root pressure generated. Then the water moves up the xylem, which we just talked about, and then out of the leaves. This whole process is called transpiration, and we can measure it with a photometer. Some leaves have slight adaptations to kind of minimize or maximize transpiration. So xerophytes and hydrophytes have slightly different leaf structure. And then finally, we talked about the phloem, the specific mechanism of loading, which is kind of H+, and then co-transport of sugar back in, and then this idea of mass flow. Do also need to know cross-section diagrams of the root and the stem and how the different tissues are arranged. So that's everything on plant transport. Hope that was useful. And then the next mind map we're going to look at this week is going to be on disease. Okay, so I'll see you then. Thanks.